Our bodies have this incredible defense network, right? The immune system. It's this super sophisticated army of cells and proteins that's always on guard against invaders. But what happens when that army is born with an inherent critical weakness? Okay, let's dive into this. And look, we're not just talking about catching a cold because you're a bit run down. We're talking about a problem in the actual blueprint, a genetic defect that compromises the very system designed to protect you, right from day one. That's where we enter the world of primary immunodeficiency. So, what exactly is a PID? Well, the key word here is primary. This means it's not something you catch or develop later on. Nope, these conditions are inherited. They're literally hardwired into your genes from the very beginning. And while any single one might be rare, there are over 350 different types. Altogether, they affect about 1 in every 1,200 newborns. And that's why getting a handle on them is so important. So, to really get what can go wrong, you've got to think of our immune system like this complex army, right? And these diseases, well, they're like critical failures in specific units of that army, from the intelligence corps all the way to the frontline soldiers. All right, first up, let's talk about the B cells. I want you to think of these guys as the body's mobile weapons factories. Seriously, their whole job, their entire purpose is to pump out one of the most important weapons we have, antibodies. See, in a healthy system, B cells grow up, mature, and just churn out these antibodies that go and tag invaders for destruction. Simple, right? But with some of these PIDs, a tiny genetic glitch just slams the brakes on that whole process. It's like the factories never even get the green light to open. And the result? The body is left wide open, without the antibodies it needs to fight off even really common bacteria. And here's a perfect example of that in action. It's called BTK deficiency. What happens here is that the defect basically freezes the B cells while they're still just trainees. They never fully mature. And get this, the gene that's messed up is on the X chromosome. That's why we call it X-linked. And it means it almost exclusively affects boys since they only have one X. The consequence is pretty clear. They get hit with one bacterial infection after another. And here's a really cool, kind of strange clinical clue for doctors. You know your tonsils? They're packed with B cells. So in these patients, their tonsils can be super tiny, or sometimes they're just not there at all. Okay, so our immune army isn't just about weapons factories. We also have the T cells. And if B cells are making the weapons, then the T cells, well, they're the field commanders. They coordinate the entire battle and even jump into the fight themselves to take out infected cells directly. You have to understand, the role T cells play is so central, so crucial, that losing them isn't just a minor setback. It's a full-blown catastrophe. Without these commanders on the field, the B cell factories have no idea what to do, other killer cells don't get activated. I mean, the entire system just collapses. The body is left defenseless against a huge range of enemies, including viruses and fungi. And that catastrophic failure has a name, SCID, or Severe Combined Immunodeficiency. This is, without a doubt, the most serious PID there is. Infants born with SCID have basically zero T cell function, and the consequences, well, they're devastating. They get hit by overwhelming infections from just about any germ out there, and without immediate, drastic treatment like a bone marrow transplant, it's almost always fatal before they even reach their second birthday. So you can imagine managing a patient with SCID requires just an incredible amount of care. Things that are totally harmless for us can be deadly for them. Take a live vaccine, for instance. For them, that weakened virus is a massive threat. Even something as routine as a blood transfusion becomes incredibly risky. Why? Because the donor's T cells in that blood can actually attack the patient's own body. It's called graft versus host disease. So what's the solution? It's actually pretty clever. All blood products have to be irradiated first. That zaps the donor T cells, making the transfusion safe. Okay, so we've talked about the weapon factories and the commanders, but what about the frontline soldiers, the grunts in the trenches? Well, that's where the phagocytes come in. These are cells like neutrophils that just rush to an infection to literally eat the invaders. So these neutrophils, they have this amazing secret weapon. After they eat a bacterium, they unleash this powerful chemical explosion inside themselves. It's called an oxidative burst, and it just obliterates the microbe. But what if that weapon is broken? In some PIDs, that's exactly what happens. The neutrophil can still eat the germ, but it can't kill it. Think about that. The invader is now hiding out, safe and sound, inside the very immune cell that was supposed to destroy it. A classic example of this is a condition called chronic granulomatous disease, or CGD for short. The neutrophils just can't make that oxidative burst happen. This makes patients really vulnerable to a specific set of germs. See, certain bacteria and fungi like Staph aureus are smart. They produce their own defense, an enzyme called catalase, that basically puts out the weak chemical fire the neutrophil can still manage. This lets them survive and cause these really nasty, deep infections and abscesses. Okay, so we've got all these different ways the immune system can break down. The question is, how on earth do doctors figure out which specific part is broken? This is where things get really interesting, where the clinic turns into a detective agency. And just like any good detective story, the investigation follows a logical path. You start broad, with some basic screening tests. A simple complete blood count, a CBC, might show that certain immune cells are low. Or measuring antibody levels could point you towards a B-cell problem. From there, you get more and more specific, until you can literally pinpoint the single genetic typo that's causing the whole mess. And one of the coolest, most powerful tools in the detective's kit is something called flow cytometry. The best way to think about it is like a super high-tech, high-speed census for your blood cells. It uses lasers to count millions of cells, one by one, identifying them by specific protein markers on their surface, kind of like jerseys on a sports team. So all B cells wear a CD19 jersey, and T cells wear a CD3 jersey. If the machine runs the blood and comes back saying, "Hey, I can't find anyone wearing the CD19 jersey," well, you've got your diagnosis for something like BTK deficiency. They're just missing. But what about the really severe stuff like SCID? Waiting for a baby to get sick is just too late. The damage is already done. The solution? It's been nothing short of a public health revolution, screening every single baby at birth, before they even have a chance to get sick. So how do they do it? I mean, how do you screen for something so complex in a newborn? Well, the test is absolutely brilliant. It's genius. See, when your T cells are developing, they kind of rearrange their DNA. And in the process, they snip out these tiny little circles of leftover DNA. These are called TRCs. A healthy newborn's blood is just full of them. But a baby with SCID, they never made T cells in the first place. So when you test their blood, there are no TRCs, none. Their absence is a massive, flashing red light that tells you the T cell system isn't working. It's an absolute game changer. And all of this brings us right back to that number we saw at the start, 1 in 1,200. That's how common these diseases are when you group them all together. It just hammers home why awareness, and especially this kind of widespread newborn screening, is not just important, it's life-saving. So it's pretty incredible, right? We've seen how science can now track down these tiny, devastating flaws in our genetic code. We can even screen for the worst of them from the moment a child is born. The technology is here, it's real. The challenge we face now isn't a scientific one. It's about access. It's about fairness. So the question we have to ask ourselves is this: how do we make sure that this simple blood test and the chance at a healthy life that it represents gets to every single child, no matter where in the world they happen to be born?